Chapter 22, Topic 22.1, Microorganisms on our Planet. The topic objectives for this topic are: be able to define microbial ecology, summarize why our view of the abundance of microbes on Earth has changed in recent years, outline the basic process used to perform metagenomic analysis of the environment, diagram the hydrologic cycle, discuss what metagenomic sampling of oceans has revealed, and define eutrophication and discuss its consequences. This chapter covers two areas of study within microbiology: microbial ecology, or the study of microbes in their natural habitats, and applied microbiology, which is the study of the practical uses of microbes in water and food processing, industrial production, and biotechnology. Personally, I'm hoping to interest you in other possibilities that are available within the broad field of microbiology. Hint, hint. In this topic, we will be discussing environmental sampling in the genomic era, soil microbiology, deep subsurface microbiology, and aquatic microbiology. Up until recently, our understanding of unseen life has been limited to those species that can be grown in the lab. Advances in biotechnology and molecular biology have made it possible to study all sorts of stuff, from our own genome to the vast array of microbes that we haven't figured out how to grow in the lab. Metagenomics is an even more recent advance. This is the process of sampling the genes of all the organisms in a sample, be it a sample of water, soil, or human feces. That way we can analyze all the organisms, even if we can't grow them in the lab. From metagenomic studies, we have discovered that we can culture only 10% of the microbes that are in any sample. By the way, that 10% includes the fastidious, hard-to-grow microorganisms. So, how do we perform metagenomics? There are two methods covered in the textbook. This figure goes over the formation of gene libraries from samples. The textbook covers the techniques used to create gene libraries in the chapter on DNA techniques. This is a good method if you want to screen everything. You catch microbes that are very different genetically from anything that we are aware of. If you're looking for new relatives of microbes you've been able to culture, or that we have information on from previous studies that made gene libraries, you can use PCR to find specific gene sequences. Often, we use primers from conserved genes, such as ribosomal RNA, or rRNA, that lead to cell death if the sequence varies very much. So, if I'm studying relatives of E. coli in cow intestines, I use a primer for the 16S rRNA genes to catch similar genes. Then I can amplify them using PCR, or polymerase chain reactions, to make enough copies that I can study them to determine the difference between the bacteria I'm familiar with, E. coli, and the unculturable relatives. This is a hot field, and you may want to consider getting the education needed to become a part of the exciting advances in this field. It used to be, to study microbes in the soil, you took a sample, shook it up in water, then viewed the results under the microscope, and/or did a plate count to determine numbers of different organisms. Soil microbiologists still do a lot of this, but the field has changed a lot. We are now interested in how microbes interact with each other and with their environment. You're probably thinking, yeah, how can they interact with their environment when it's just dirt? Well, think of soil on the level of the microbe. There are microscopic pockets and sand grains that are entire worlds to a bacterium. I really like this figure because it shows this dynamic, varied world. A plant rootlet is huge, cutting across the soil and providing habitats for bacteria and fungi that live in association with plants. This pocket has areas that are dry, like there, and areas that are submerged, like here. It's an entire ecosystem with sideways and upside down lakes. Each area has its own complement of microscopic life that interact. Bacteria and fungi break down dead matter. They release organic matter for other bacteria, which produce necessary metabolic products for other bacteria. Protozoa, nematodes, and invertebrates feed on the bacteria on each other. It's the circle of life in an area the size of the period at the end of a sentence. So, why should we care? In the end, it's just dirt. Well, do you like to eat? Plants wouldn't grow in their staggering variety if not for soil microbes. If not for soil microbes, the leaves you mulch in the fall wouldn't turn into fertilizer for your garden. Don't believe me? Try mulching ginkgo tree leaves. They have molecules that soil bacteria and fungi are unable to break down very fast, which leaves you with a whole bunch of leaves in the spring, pretty much the same number as what you had in the fall. Since soil microbes naturally specialize in breaking down a wide range of molecules, applied microbiologists are currently working on growing them in the lab, so we can apply them to polluted sites. They are also working on figuring out how to stimulate the growth of specific bacteria in polluted areas to speed up cleanup. They are working on how to use microbes to clean up sites such as the Fukushima nuclear reactor sites that were damaged during the 2012 tsunami. We used to think most life on Earth lived at or near the surface of the planet. Well, and there's the aquatic microbes, but we'll talk about those later. It makes sense, since the sun provides most of the energy on the planet. But, using metagenomics, researchers are finding a lot of microbes deep under the surface of the planet. Of course, there are a lot of microbes living in caves, like this one shown here, but there are a remarkable number living deep in solid rock. Some researchers are estimating that most of Earth's life lives in the deep lithosphere. How they are doing this is being researched. Since there isn't much organic material, no light, no oxygen, it's cold, and there aren't spaces to wiggle in like the topsoil provides. And do they affect life on the surface? Do they model early life? Will we find early life forms that have continued to survive buried deep in the earth, like living fossils? Further research will tell. Aquatic microbiology has been surging ahead, too. We will discuss applied aquatic microbiology in the next concept, but let's do an overview. Let's go over the hydrologic cycle. Surface water evaporates from heat, respiration of aerobic organisms, and transpiration of plants. This gaseous water condenses into clouds, which forms precipitation such as rain or snow, where it falls back on the surface. The precipitation ends up in the ocean, freshwater lakes, and streams, and so on. Most of the surface water flows back into the ocean or is evaporated back into the air. Some water ends up in the deep earth as aquifers. Microbes are found in all of these environments. We thought the air was pretty much microbe deficient, but recent studies have shown that bacteria are responsible for much of cloud formation. Soil bacteria are kicked into the air by winds, and water condenses on these bacteria. Think about that the next time you're standing out in the rain. Vast populations of microbes, including viruses, have been found in every aquatic environment. Their numbers and diversity are affected by temperature, nutrient levels, salinity, and so on. They are adapted to the environment they live in. Lately, we humans have been interested in how our activities throw things off. We like to think of fresh, flowing water as being sterile, but it is full of life. Generally, though, the numbers of cells per mil are low enough to prevent the water from being turbid, or cloudy. That would be anything less than 6 million cells per milliliter. Of course, when water is still, microbes stay in one place when they reproduce, producing turbid water like this pond. Algae and cyanobacter form floating mats and green-slimed biofilms on submerged rocks. Fertilizers that run off from agricultural fields can cause these microbes to grow even faster. Remember, fertilizers contain lots of nitrogen and phosphorus for plants to grow, and microbes need the same macronutrients, like how I worked that vocabulary term in from the chapter on microbial growth requirements. When these microbes grow very fast, or bloom, they use up the oxygen in the water as a part of the normal aerobic respiration process. The oxygen that they produce from photosynthesis replaces some of this, but most of the oxygen escapes into the atmosphere. This is all fine and wonderful for us air-breathing humans, but fish and other aquatic life often die off. Also, some species of cyanobacteria produce neurotoxins that have been linked to increases of neurological disorders in communities that use the water that have had algae blooms in their water supply. Marine protozoa, called dinoflagellates, also experience blooms. They do this naturally, but there is some evidence that fertilizer runoff can cause them to bloom more frequently. Some dinoflagellates have red photosynthetic pigments, and when they bloom, they turn the water red. Some of these red dinoflagellates produce neurotoxins, which kills fish and sickens humans and other animals that eat the fish and other marine life, such as clams and mussels, that have eaten the dinoflagellates. There have been research into tracking red blooms, or red tides as they are called, so that affected populations can avoid eating affected seafoods. Also, research is being conducted to help farmers apply only the fertilizer their crops needs. This helps the farmers save money and it helps the environment. Everyone wins. Well, that's it for this concept. Here is a reminder of the learning objectives for this topic. Be able to define microbial ecology, summarize why our view of the abundance of microbes on Earth has changed in recent years, outline the basic process used to perform metagenomic analysis of the environment, describe the hydrologic cycle, discuss what metagenomic sampling of oceans has revealed, and define eutrophication and discuss its consequences.